The overall goal of this imaging approach is to monitor the nitric oxide signals in real time on the level of individual cells using a novel class of genetically encoded fluorescent nitric oxide probes, the geNOps.
Considering the importance of nitric oxide in biology and physiology, a visualization of this molecule in cellular or even subcellular level has been desired since it was discovered.
We visualize the nitric oxide signals with novel, rather simple-built fluorescent protein-based probes, the geNOps.
The geNOps respond directly and immediately to nitric oxide by fluorescence quench. This means you can monitor the real-time kinetics of the generation, diffusion and degradation of nitric oxide in single cells.
The main advantage of geNOps is that you actually can look at nitric oxide signals with high spatial and temporal resolution using a conventional fluorescence microscope.
Begin this procedure by replacing the storage buffer of the endothelial cell line or HEK293 cells with 1 ml per well of the iron 2 booster solution at room temperature. Incubate the cells for exactly 20 minutes in darkness.
The iron loading procedure is essential to get full responsiveness of the probes. It might be adapted for different cell types and applications.
Afterward, wash the cells once with storage buffer and incubate each well with 2 ml of storage buffer for at least 2 hours at room temperature in order to allow the cells to equilibrate. Then, replace the storage.